Hello guys, so this is our pitch for Intel Python hackfury 2. Okay, so the problem which we are solving here is protein interresidue distance prediction. So what is this problem? The problem here is to, to predict the distance between two residues of protein. We will be explaining it shortly. Okay, so as I have stated, the protein folding challenge or the problem which we are solving is important, but one should not believe on the words. Okay, so why it is important is this. Okay, so all the constituents of the cells of whether it being a human cell, a synthetically created protein, or be it any from any living or non-living organism on the boundary. For example, viruses are on the boundary of living and non-living. Okay, so to understand how these proteins which make these cells interact with any other biological entity, whether it be drugs or some other physical thing or physical living organism, whether it be viruses or other proteins in the cells, we need to understand this protein folding. Okay, in short, what we can do is this, that if we are able to solve this protein folding problem, okay, we can control aging, we can create new species like superior breed of humans, superior breed of some animals, superior breed of plants, okay, control any disease, whether it be HIV or cancer. So in short, we can be very close to addressing a lots of problems human generation is going to see or is already seeing. Okay. So coming next is what is the protein folding problem. Okay. So these proteins are made from essentially carbon, hydrogen, and other nitrogen elements. Okay, so what happens is that when this particularly amino acids which make these proteins, okay, how they interact or how they coexist very close to each other, the distances could be in angstroms. Okay, this is what is the interesting part which makes the functioning of protein unique with their structure. Okay, so the biological function of a protein is intimately linked to its 3D structure. Okay, and the prediction of the 3D structure is a very difficult task. Why? Because we can't simply probe into the cells and see how it is. Why? Because whenever it is done, we have already disturbed the cell structure. Okay, coming next, so this is how it looks. Okay, so this is so here what on this side we have this amino acids. Okay, this is a strand of amino acids which when folded together it becomes like protein folding. Okay, and amino acids are basically responsible for making these proteins. Okay, so the left image is before folding and the right image could be called after the 3D folding has happened. Okay. Now, so this problem seems to be interesting, but the obviously the question arises is that are we? Is the problem new? Are we the first person to formulate this problem? It is not. Actually, the protein folding problem has been there since 50 years. Okay, and believe me, people have been trying to solve it very thoroughly, and there have been improvements. Okay, for example, DeepMind's AlphaFold model recently made into the list of millennial innovation for innovation list, and in the last decade there has been a lots and lots of research on the protein folding problem which have employed lots and lots of new techniques, for example the deep learning and machine learning for prediction of the 3D structure of the models. Okay, so our problem, the approach which we are taking to solve this 3D structure prediction for the protein is from is motivated by the deep learning methodology. So how we are we are going to address this problem or approach this problem? So what we are going to do is this. Conventionally, people have tried this, that they take a protein sequence, which is essentially amino acid sequences. Okay, so they see this particular structure and try to match it with some known structures, okay, which they have already enlisted in certain dictionaries. Okay, so this dictionary have been made with a very difficult experimental procedures, so it is not always feasible that we make that for every protein which we encounter we make that experiment run and understand its 3D model. Why? Because it is very experimental and it costs a lots and lots of money. Okay, okay, so moving next, what we are going to do is this, that what if we don't have such templates for proteins? Okay, so what we do is that we have to work without having certain templates which already matches with certain other proteins which for which the 3D structure is known. Okay, so such prediction is called ab initio prediction. Okay, in such cases what we have to do is this, that the contacts between two residues of the protein, the residues are essentially the outgoing nodes of the chemical compound which interact on the edge of the protein. So if we are able to predict the contacts in the proteins, it would be very crucial, and they serve as like anchor points for generation of 3D model of the protein. Moving next, what we can do is this. So essentially I have mentioned that, okay, so we want to predict the contact between proteins, and we are going to model the 3D structure from there. Okay, so essentially what we are going to do is this, there are two residues and we want to predict the distances between this. If you are going to take this for entire in all number of residues in the protein, it is going to be called we call it inter-residue distance matrices. Okay, and this is essentially distance between two residues. So what we are going to do is this, that we are going to have a fully convolutional neural network and formulate this distance map prediction as a regression task. This regression task is very important, and we realized that this mitigates a problem of class imbalance if we were going to approach this problem using classification, which people have done in the literature. So this is one of the novelties which our solution has with respect to the existing literature. So coming to the data set, we have the data set which contains around 3000 proteins and 6 features for each of them. So these 6 features are not essentially images, as we discussed, we showed that we will be using CNNs for this prediction. Okay, so these are not essentially 2D images, but CNNs are meant to work on the data on the grid. Okay, whether it be 1D or 2D. Okay, so what we do is this: we convert these 6 features into 13 images, which are going to be the input to the model. Okay, so how does it look? So here we are having the input images. Okay, and this guy here is the predicted distance map. Okay, so given this all 13 entries, we want to predict the last one. Okay, this is the inter-residue distance matrix. Okay, realize the fact that these two guys, the first one and the second one, are transposed to each other, and similarly it happens for 5 pairs. So what we do is that we convert this, we use this symmetry property to our benefit and convert this for these two channels into one channel image, which is the finally here, and that results in dimensional reduction of the input. How we are going to evaluate our performance, the CNNs which we are going to formulate as a regression task, and we are going to predict the interresidue distances. So there are three metrics which is known into the literature. It's called mean average error, so this is called mean absolute error also. Okay, so mean absolute error of top L long range. So this is a new term which is coming in. Another thing is average precision. Okay, and then something called TMS score of top one model. Okay, so this is from the literature. So what does it essentially the first metric captures? It captures point wise mean absolute distance between the predicted and the actual ground truth of the interresidue distance matrices. Okay, the next metric was capturing the precision of long range contacts. Okay, so there are three kinds of contacts, interresidue contacts. It's called short range, medium range, and then long range. Okay, the short range are not very critical for short range is something which is less than like around 6 angstrom. Okay, it is not going to be critical for our analysis. Okay, 3 to 6 angstrom I should mention. Okay, and it is not going to be very critical for the 3D model generation as people have shown in the literature. Okay, the medium range is again not very important. What is very important for the 3D structure generation of the protein is the long range contacts, which are at least 24 residue apart. Okay, when I say residues, again I just like to remind that the residues are just chemical elements on the periphery of the protein which are responsible for any reaction. Okay, so the loss function which will be using for our training is MSE loss, mean absolute error, and binary cross entropy and symmetrical loss. Okay, the symmetrical loss and binary cross entropy is two interesting innovations which we have proposed. Okay, so binary cross entropy, what it does is that in our prediction, what we do is this, that we predict that okay, certain distance between two residues is whether it is greater than 8 angstrom or less than 8 angstrom. This provides us an additional benefit on the proposed metrics. Why? Because then we are just asking the model to say that okay, you can't be wrong by predicting more than 8 when it was supposed to be less than 8, and the rest of the losses are going to penalize it how far we are predicting from 8 angstrom. Okay, or the actual ground truth. The symmetrical loss ensures that we are going to predict the distance matrices, which is again a symmetric matrix. Why? Because the distance between the two residues, irrespective which you count first. Now the architecture which we are using again is a pre-trained network from ImageNet, and as this we have said earlier that this is fully convolution network. Okay, we will be using a VGG unit, where the VGG comes from ImageNet pre-trained weights are there, and the unit structure as shown on the screen. So this is also one of the innovations which we have proposed. No one earlier have used pre-trained networks. Why? Because they were not sure that whether ImageNet features are going to be important for protein residue contact prediction. Now this coming to the results, we achieve average MAE of 2.45 angstrom and average precision of 0.99, which is remarkably good. Okay, and average TM score of 0.31, which is not very good, but given the fact that we have not been using this structural mapping which we discussed earlier, we have to do it blindedly and just using the residue contacts. It's very good. Okay, the standard literature says that it will be somewhere from 0.21 to 0.4. Okay, so we are well in the margin of the literature. Now just presenting few of visual results. So this is the ground truth, and this is our predicted map. Okay, and this is the less than 8 angstrom, and other one less than 8 angstrom, and this is more than 8 angstrom. Okay, and this is a corresponding prediction for that. Okay, on part of Intel libraries which we have used, these are the Intel libraries we use. Okay, so this on the screen we have displayed the Intel libraries which we have used for developing our project here. So obviously Intel NumPy scales very good with the Intel processors, whether it be Xeons or Core i7 processors which we have been using. We don't have access to Xeon, so but these libraries are still feasible for with i7 processor. Okay, and apart from that, Intel SciPy was also relatively faster than the other GPU enabled things on and the SciPy functions which run on the GPU. Okay, Intel Math kernel was used for deep neural networks, and we also use an Intel optimized PyTorch framework for training our deep learning framework. Okay, a deep learning architecture, and obviously we use MATLAB and Jupyter notebooks from Intel Python distribution. Thank you.